All right, CRISPR, the wonderful gene editing technology that usually, but not quite always, gets it right. Let's learn about CRISPR today. And so CRISPR stands for clustered, regularly interspersed, short, palindromic RNA, CRISPR. You can see it written right up here if you want to have a look at that. There we go. And that would be a good thing to include in your answer. And so, what CRISPR actually is kind of an immune system for bacteria. Now, bacteria are single celled creatures. They don't even have a nucleus, so they can't really have an immune system like we would think of it. But on the other side, the only thing that they're going to have to worry about really is attack by viruses. And there are a lot of viruses out there that are trying to destroy bacteria. Most of these viruses, in fact, nearly all of the viruses that infect bacteria, have double stranded DNA as their genome. And so, the first thing that happens in an infection is that this double stranded DNA is going to be injected into the cell. And from this point, the bacterium has about 15 minutes before it's going to be dead, essentially. And so, it needs to shut down the virus's DNA before it can do anything, before it can replicate or do any of the things that it does. And so that's what the CRISPR system is. And really, we call this the CRISPR-Cas system, or CRISPR-Cas9, CAS9. Because this is how it works. It is a set of these clustered, regularly interspaced, short palindromic repeat RNAs. It is an enzyme that will use these things in order to do something. So, first of all, here's our CRISPR array. So you've got all these genes up at the front. They're going to make the Cas proteins, the CRISPR array proteins. And so in this case, our Cas9 equivalent, or here's called Cas3 in this kind of bacterium, is going to be made. And what it does is it cuts double-stranded DNA. But of course, the bacterium doesn't want to cut its own double-stranded DNA. It wants to make sure it cuts specifically virus double-stranded DNA. And so what a CRISPR array is it's little pieces. And these little pieces correspond to different viruses that this bacterium commonly encounters. And the viruses actually build these up. These are kind of like a record of everything that the particular bacterium has defeated, in its evolutionary history. Some of these bacteria have great big arrays of these. Some of the viruses have since gone extinct, or at least haven't been discovered yet. And so you have these cases where the CRISPR array is the only the last remaining record of some of these viruses from the past. It's kind of like a molecular fossil, which is sort of neat. So anyway, the way our CRISPR-Cas system works is that we've got a little piece of RNA that we're going to make. And the little piece of RNA is going to serve as a guide. We're going to, the guide is going to lead the Cas9 protein to cut virus DNA at a specific place. And it's going to be set up in such a way that it's only going to go after sequences that appear in the virus genome. The way it does this is that it's not just going to read out these little genes, because, if it was just cutting wherever it had this little gene, then it would cut its own DNA. And the bacterium doesn't want to do that because that's bad. But what it does is it actually folds up this DNA. And if it's not folded up in just a perfect way, then it doesn't work. And when it's folded up, the bit that matches the virus actually comes from two different sections. And so with these two sections, it then can be specific and it can just knock out viruses. And so most kinds of bacteria, including all the ones in your gut, are going to come preloaded with something like a CRISPR and Cas9 system. This is just how bacteria are able to survive in the world, and part of how they do business, how they live in a complex world full of dangers. So, the word CRISPR, this comes from the array. So the CRISPR is actually this little set of little tiny pieces of viruses that we're going to copy And then the Cas9 is going to be the protein that uses one of these little pieces. And here the little pieces are all chopped up into little CRISPR RNAs. The Cas9 is going to hold on to this and use it to basically line up a target on a piece of viral DNA. And as long as it is the same virus or with the same sequence as this cell has defeated before, then it's going to be able to reach that double-stranded DNA and cut it. And at least break it in half. And if you break a virus in half, break its genome in half, usually you have killed that virus. There's no way for it to really hurt you after that. Then the other neat part about it is that the enzymes, after they are done cutting up a virus, will grab onto any leftover viral DNA. And so the bacterium is able to tell viral DNA from its own DNA because it puts a lot of methyl groups onto its own DNA. And so it's kind of marked its DNA. So it's got the regular nucleotides, ACGT, but they've got extra little methyl groups attached to those, some of those anyway. So it can recognize when a piece of DNA is not methylated. That means it's come from a virus, because this is something that bacteria do and viruses do not do. So when Cas is going to grab onto one of these unmethylated pieces of DNA, there are a couple other of these Cas proteins. They're all going to get together and they're going to chop up the DNA into little pieces, and they will pick up pieces just at random and take them down here, make a little piece of DNA copy, and insert it at the end of the chain. And so every time the cell defeats another virus, it makes a little mark. It adds a little tally to its stack of CRISPR sequences. So you'd say, okay, this is kind of neat. This is a little bit of fun with bacterial genomics, but what is the relevance to the wider world? Well, it turns out that if you can control this little part right here and make that bind to any specific piece of DNA, then as long as you have a Cas9 protein in the vicinity, it will grab onto your little piece of RNA and it will line up on whatever that piece of DNA is, and it will cut it. And so this is a way to cut DNA inside of a living cell. And if you can cut DNA inside of a living cell, that is the first step to being able to change DNA inside of a living cell. And so that's why this was the Nobel Prize. Jennifer Doudna and Emmanuelle Charpentier are the ones credited with... They didn't first discover CRISPR, but they turned it into a practical tool that can be used in mammalian cells to modify DNA while the cell is still living. And modifying DNA is the step that allows us to potentially start controlling our genome at will. So you may have heard there was already at least one case. A scientist in China genetically modified the DNA of, I believe it was twin girls, that were then implanted. The mother had the babies, and sure enough, they carried the genetic markers that the scientist had put in there. And then after that, of course, he disappeared and stopped doing science, and is believed to be under house arrest, last I heard. So, yeah, there are risks, certainly. I don't know. If we were in class in person, I would say, how do you feel about this? What are your feelings on genetic modification? Because it's got potential to do a lot of good. Genetic diseases, certain kinds of muscular dystrophy, for example, certain kinds of coagulation disorder, can be cured once and for all, and you can actually pass the cure on to your kids. But if you do anything weird in your DNA, you can also potentially pass that on to your children. And so the changes that we make could be forever changes. And after this lecture, so we're just going to cover CRISPR here. This is kind of a two-parter. We're going to go to a thing called a gene drive. And a gene drive is a CRISPR-Cas9 system that will jump from one chromosome to another and from one individual to another, like an infectious CRISPR, kind of, yeah. Only not, not the same way as a virus is infectious. So stay tuned for that. That's going to be, I think, very interesting. Okay. So what does the CRISPR look like? And remember, the CRISPR is the little bank of genes. And so what it looks like is a set of clustered regularly interspaced palindromic repeats. So palindrome is a word that means something that reads the same forward and backwards. So like the name Hannah right here, Hannah or Hannah, like that. Or a man, a plan, a canal, Panama, something like that. That whole sentence is a great big palindrome. I don't know. Every year when I would ask in class, people tell me new ones, and yeah, they're pretty cool. I think race car. Yeah, that works. That's also a palindrome, I believe. Anyway, so palindrome in DNA is actually a reverse complement palindrome. So what you'll have is GTTTT, GGAAC right over here, and you will have on the other strand GTTT, AGAGC over here, which is, I guess, a little bit different, but pretty close. But what we would actually see if we were looking at just at one strand would be kind of the opposite of this particular strand. So, and this is an imperfect palindrome, but it's pretty close. And so when we copy this one, then we're going to get nice little, there we go, piece of RNA with the same sequences on there. And the thing about a palindrome is that it can fold up to make a shape that is called a hairpin. So this little thing down here, kind of looks like an upside down lollipop, is a hairpin. And it's just the C on this side sticking to the G on this side. If you have a palindromic repeat where the same DNA or RNA, in this case letters, go backwards at the different part of the RNA, then what you will see is it will fold up into a little structure like that, which is neat. And so all of the CRISPR sequences have a unique region in the center and a palindromic region on the outside. And the bacterium will take this thing and it will chop it into just the right size pieces. And then what it does is it's got this little loop up here that it's going to hold on to. So this is a thing called the guide RNA. And the guide RNA is going to have a little part down here, and this is going to be complementary to whatever sequence we're going to cut. And then the Cas9 enzyme will just grab onto this guide RNA with its little hairpin, just like that, and it will hold onto a piece of double-stranded DNA, scan along, and if it finds something that matches the RNA down here, so if it's a reverse complement of this RNA, then it's going to recognize it, and it's going to stop, and it's going to cut it at a site that's exactly right here. And so you can make a guide RNA to cut a piece of double-stranded DNA, like a chromosome, at any position that you want. And that is all CRISPR-Cas9 is. It is a way to cut DNA. And you'd say, well, cutting DNA, that just sounds like DNA damage, and that doesn't sound like I'm going to be able to incorporate other genes into a living cell or delete genes or whatever. And yeah, you're partially right. All of this changing when we add or subtract genes is going to happen at the next step, when the cell tries to repair the DNA break that you made. So the point of CRISPR-Cas9 is just that it makes a double-stranded DNA break. And then the cell has got a problem, and it's got to figure out a solution. And what you do if you're designing a CRISPR-Cas9 system and you want to change the DNA inside of a cell, is you offer the cell a clever but kind of tricky and a little bit false solution. So you give the cell something that it's going to think is an adequate solution, but secretly you know that you're actually manipulating the cell. You're actually playing the cell. So yeah, let's look at how that's going to work. All right, first of all, this is the Cas9 complex, and you can see it here in complex with the guide RNA and the target DNA. So the sgRNA is the small guide RNA, so just think of it as the guide RNA. And so here you can see it snaking up here, wrapping around there, coming down here, and that's fine. This is the part that's double-stranded and looped around. So this is the palindromic part up here. The DNA that we're going to cut is right here, and you can see this one has already cut the DNA. There's one end of it, and the other end would be right here. Yeah, we've broken it off already. Originally, the DNA would have extended further down here, and then this is the point at which it gets cut. So at a certain position in the guide RNA, that's marking the cut site. And you can look this up, or there are little websites that'll design a guide RNA for you to cut anything that you want. So, like I said, that is all the CRISPR-Cas9 system does. It cuts DNA, and then it leaves the cell with a problem. How do we fix this DNA? Because if the DNA stays broken, the cell just dies. And so this is a risk. Some of the time when you cut the DNA with a CRISPR system, you will end up with a dead cell on the other side. So this is not the sort of thing you want to just routinely do, at large scale inside of a living organism, because if you lose too many cells, then yeah, that's not going to be good, right? It could cause more damage than whatever the problem is that you're trying to repair or alleviate. So we cut it, and then there are two ways that a cell can repair. One is the way that it will repair if it does not have anything to copy, and so that's called non-homologous end joining, or NHEJ. The other is the way that the cell will repair itself if it does have something that it can copy, and that's called homology-directed repair. And so I think we're going to look at, both of these things on the next couple of slides, but these are the main ways. So homology-directed repair is the main reason why you have 2 of every chromosome, except potentially, if you're male, you only have 1 X chromosome and 1 Y chromosome. And if you're female, you have 2 X chromosomes, and so you've got a backup for everything. And good job. Yeah. You won the genetic lottery. Yeah. That's just how it is. Right, but first let's look at homology-directed repair, because this kind of makes sense. So the homology means an exact DNA match at the spot before the break and after the break. And so what the cell is going to do is it's going to line up the ends here, the blue parts here, and it's going to line up these blue parts, and it is then going to copy whatever is in between the blue parts, and it's going to repair the DNA. So even if the DNA has lost several nucleotides, even if you had a whole chunk that went missing, you can then repair it, and you can build it right back in. The other alternative is non-homologous end joining, and non-homologous end joining is chaos. So in this, what the cell will do, if it doesn't have anything that it can copy, so for example, if you have a break in your Y chromosome, there is not going to be a second Y chromosome there for you to copy, because that just doesn't work out genetically. And so, you are going to be stuck with the cell trying to figure out a way to glue this DNA back together. And if there's no template to copy, the cell basically has no idea how to do it. And so it will randomly add and delete nucleotides until it gets a couple nucleotides on either end that actually are able to base pair with each other. And then once it has that little base pairing, it will then fill in the rest of the gap and repair it. And so you will either be deleting or inserting random nucleotides at a position in a gene. So if your goal is to disrupt a gene to see what the gene does, for example, then CRISPR is a great tool. You just set your CRISPR to work. You cut both copies of the gene if there are more copies there, and you just let the cell try to repair it. You will get a gene with either more nucleotides or fewer nucleotides than it should have had. And most of the time, this is going to end up causing a stop codon in the gene, or changing what the gene actually codes for. Because remember, we don't read DNA in single nucleotides. We read it in groups of 3 nucleotides. So if you add 1 nucleotide in the middle, you shift off all the other sets of 1, 2, 3, 1, 2, 3. And so none of those are going to be read as normal again, even though they're right there. And so you'll end up making the wrong protein, or no protein whatsoever. And so that's one way to get rid of a gene that you don't want. A way to add a gene that you do want is through homology-directed repair. So what you can do is you can break one or both copies of a gene, better to break both copies, and then after both of them are broken, you actually put in a fake piece of DNA that you've made in a laboratory that matches everything on this side and everything on this side of the gene, but has something extra in the middle. And if you put this in there, about 4 times out of 5, the cell will line up the bit before the break, the bit after the break, and it will then fill in on its own chromosome whatever you tell it in the break. You can put an extra gene there, you can put an extra 3 or 4 genes there, it's fine. You can, in this way, transfer genes into the DNA of an organism while it is still living. And that is a powerful technology for just investigating biology and understanding how living things work. So, let's have another look at this. In homology-directed repair, which I think is the desirable outcome in general, if you have a template that you can copy that is the same gene as the one that got broken, then even though this is broken, a double-stranded DNA break, you can actually pair up with the 2 strands of the copy, and you can fill in any little gaps in there, and so you can end up repairing the chromosome. That's homology-directed repair, and that's generally the better outcome for the host anyway. The other version of this is, of course, non-homologous end joining, and so that's going to be shown over on this side over here. Homology-directed repair, we're showing that again over here, where you're copying a template and just making a good copy of it that fills in both ends of the thing that is broken. In non-homologous end joining, we are actually going to use a separate set of enzymes that will add on. So they will add on nucleotides and they will take away nucleotides. And then when we have enough that we can get them to stick together a little bit, a thing called ligase 4 is going to come in and actually join them up. At least in a human, that's what the genes are called. So yeah. Now the problem is, when you do CRISPR-Cas9, you cannot be sure, even if you put in a template for the cell to copy, you cannot be sure that that template is going to end up in exactly the right place at exactly the right time to be the template for repair. Some of the time, you are going to end up doing non-homologous end joining even when you would intend to do homology-directed repair. And so it ends up being something like 4 out of 5 times you'll get homology-directed repair if you do this right. But that 1 out of 5 times, yeah, you probably end up with non-homologous end joining. And this can ruin whatever gene you are trying to manipulate. And can potentially be lethal or can carry a mutation that would then be passed on to the offspring of the organism. So that is the risk. All right. How can we use this in people? So 2015 is actually the first published attempt to genetically modify a human embryo, and it was actually for a very good reason. So researchers were trying to fix the mutation that causes beta thalassemia, which is a mutation in the beta chain of hemoglobin. So hemoglobin has 2 alpha chains and 2 beta chains and one of these little heme groups that kind of sits in the middle of each of the chains. And the heme group, remember, looks a little bit like the top of the chlorophyll molecule. It's porphyrin ring, same kind of general molecule, I would say. All right, and we're going to use this to carry oxygen, but also heme groups are involved in a lot of electron transport that goes on in your cells. A lot of the proteins that are connected with respiration are going to involve these. Some of those are going to be called cytochromes. And so the idea was that you have these people naturally. And when they have one mutation, you'll get a beta thalassemia carrier who doesn't have illness, but if they have babies with another beta thalassemia carrier, then you can end up with a child who is homozygous for beta thalassemia. And this person will have hemoglobin that essentially the beta subunits don't really work right. They misfold. And so you really only have functional alpha subunits. And so your hemoglobin only carries about half as much oxygen as it should. And so this person would have effectively severe anemia Yeah, and would have problems staying fully oxygenated as a result of this. And so the idea was, well, what if we could go into an embryo? Let's say we have two parents who know that they are beta thalassemia carriers. What if we could change the beta thalassemia beta chain gene and fix it so that there was no chance of passing that on to the baby? Would this actually potentially be useful? And yeah, the answer is yes, if you do it right. So here are some results from that paper, and I think I linked to the paper in the, there we go. It's going to come up down here. Right, so when the researchers were trying to mutate the beta thalassemia mutation, it turns out that you actually have several copies of the globin gene in your chromosomes. And so on chromosome 11 or 3 or 1 or the X chromosome or chromosome 6 or different parts of 1, 11, 14, and 20, you do have copies of globin genes. And these things are similar but not quite identical to the globin gene that's involved in hemoglobin, the beta globin that's going to be the hemoglobin beta chain, HBB. And so they put in a little guide RNA sequence that was specific to this, hoping that these were enough changes that they wouldn't mutate any of those. And so here we go. We got, let's see, 11 out of 11 and 17 out of 17 mutated. Fantastic. And with this other one where we had 3 mutations there, none. But, uh-oh, look at this one. For the one on chromosome 11, the other one at a different place on chromosome 11, they got 4 out of 10 and 2 out of 10 mutated. For this one on chromosome 6, they got a total of 10 out of 23 mutated. And no other changes here, but look, that even has 4 mutations in it. So the CRISPR-Cas9 is pretty good, but that guide RNA, the little RNA that tells Cas9 where to cut, doesn't have to be 100% matching to the sequence that it's eventually going to cut. Occasionally, if you have enough CRISPR and Cas9 around, you will end up cutting sites that are close but not quite the same as your intended target sequence. And so as a result, here's what you end up getting. So they showed that the embryos, they were targeting these at a fairly early stage. These are actually being targeted at S phase of the 1-cell stage, so that's pretty darn early. And what they found is that when they put this in there, they eventually got a large number of embryos that they managed to change. So over here, they managed to change, let's see, oh no, this was 50-50. And so this is looking for the mutation in the beta globin gene. And with the CRISPR-Cas9 working, they actually managed to get, instead of just 47% of the genes being wild type and 52% being mutant, they actually managed to bump that up to 66% that were fully wild type, fully repaired, only a little less than 10% that were still mutant, which is pretty good. And 24% it says is mosaic. What that means is that they were able to repair one chromosome, but the other chromosome still had a problem. So yeah, that's what a mosaic is in genetics anyway. Now the question is, how did they repair? So they did another one of these sets of tests, and what they found is that of the changes that they had made, where they definitely made a change, because they put a little marker in next to it to tell if it was the new version or the old version of the gene, but they put the marker in such a way that it wouldn't interfere with the actual gene's expression. A little over half the time, they got exactly what they wanted. About 20% of the time, they did not get what they wanted. And almost 30% of the time, they got non-homologous end joining in this particular experiment. Over in this experiment, they redid it. They ended up this time with about 36% non-homologous end joining. And they kept doing the experiments, but every time they'd end up with somewhere around 20 to 30% of the mutations ending up being repaired by non-homologous end joining, which would essentially damage the gene they were trying to repair. So that's not good, right? And then over here, they actually found that they ended up with quite a number. So about a quarter of the mutations that ended up going in there were mutations in genes that were not the exact copy of globin that they had targeted. These are other globin proteins that are getting mutated. So you can see that, targeting, inside of a living cell is not a risk-free process. And when you're doing something like this in the laboratory, it's relatively easy. You can just repeat this thing 1,000 times, and then you can only pick the ones where the genetic change has happened exactly as you plan it. But if you're doing this inside of a living, embryo, something like that, then it's kind of hit or miss, because you're not going to know whether or not it worked until after the embryo grows up into a little baby and you can sample it, or at least sample it at a later stage of development. And so at that point, you can't really go back and say, no. You can't just put 100 of these embryos in and hope that one of them's going to be good and just quietly get rid of the other 99. I mean, human ethics does not allow that, I'm pretty sure. So this is the difficulty with actually translating this into something that can be used for human genetic manipulation. I think that technology is still a ways off. We've got to get much better at targeting our CRISPR-Cas9 in such a way that it does not make mistakes. And there are people that are making strides in doing this, but there are still problems. So mostly at this stage, it's a useful research tool or a tool for destroying genes that you don't want. It's fairly efficient at that. It's just not as efficient at always giving you exactly the genetic outcome that you do want. Unless, of course, you're after chaos, and then it provides chaos, absolutely. What this study looked at is whether or not you could actually do this at different stages. So the experiment where they actually made genetically modified humans, they did it, I believe, at the 8-cell embryo stage. So when there's 8 cells inside of that little embryo, and you can actually genetically modify each of these cells. And the problem was that both the embryos that went on to become the little twin girls, eventually we figured out that they were both mosaic. So they had some cells out of those original 8 that were modified, some cells that were not modified. And among the modifications, they had a mix of homology-directed repair and non-homologous end joining, if I remember correctly. Yeah. So it was kind of a mess. And now, instead of this just being a laboratory experiment, it's an actual child that has to go off to school and, do stuff in the world and has parents that like her. It's a little fraught. But the idea behind this study was that if you were to go back and not be a total amateur and modify the 8-cell stage, but modify the single-cell stage, if you made the change, then the change would be carried through, because every cell that divides from this one cell is going to be genetically identical, at least at the start. Whereas if you modify at this stage, you've got 8 different experiments essentially happening inside of each embryo, and some are going to succeed and some are not. That's just the nature of CRISPR-Cas9. And so they looked and they found that actually you get really good development from if you genetically modify just the fertilized embryos at the 1-cell stage before they've even divided. You can get pretty good survival if you modify them at the 8-cell stage. So I think based on earlier experiments in other kinds of animals, they thought that the 8-cell stage was where they had to manipulate the DNA. They couldn't do it at the 1-cell stage, but that turned out not to be the case with human embryos. And they went and checked that as you let these get older and turn into a stage that they call a blastocyst, which is something we cover in biology too, your efficiency goes down to about 50%, whether or not those even survive and are able to develop. So it's really much better on the cell. If you're going to genetically modify it, you should hit it as early as possible. It's the cleanest, and the cell has the best chance of actually surviving intact with the mutation that you made. So, once again, just reviewing, remember, CRISPR-Cas9, its only function, it's got that little guide RNA, which is inside of Cas9, and its only function is to cut a chromosome. And then the next step is that you're going to have to repair that chromosome, and it's either going to be NHEJ, which is chaos, or homology-directed repair, HDR, which is the good one. Now, I'll mention this here, but we're going to cover this more in the next lecture. So this is a thing called a gene drive. And so what this is a little plasmid, and you're going to put this plasmid in, which plasmid is just a circle of DNA. And the plasmid has a Cas9 gene, so it can make a Cas9 protein. And it has a guide RNA gene, which can make a guide RNA that fits into the Cas9 protein. And we're making a specific one that will actually bind to a site on a chromosome. And so if you put this in and you boot up the Cas9 and the guide RNA, then they will make a little CRISPR-Cas9, and they will cut the chromosome. And then the other thing that makes this into a gene drive is that before the CRISPR-Cas9 part, you're going to have homology regions. So a region that matches the area just before you cut, and a region that matches the area just after you're going to cut. And so once you cut this, boom, then the idea is the cell is going to line up this bit with this bit that matches, same here, and then it's going to say, well, I must have to fill in everything that's in the middle, because the cell doesn't have any way to know or care, how much it's filling in. It just really wants to join that piece of DNA back up. And so, if you have two homology regions, a Cas9 gene, a guide RNA gene, and then we call this gene the payload gene, because this is what you're going to end up putting into the genome. You're going to make a genome that actually is going to have a CRISPR-Cas9 system built into it. And the neat thing is that if you have one chromosome that has this in there, it's going to make the Cas9 protein. And it's going to make the guide RNA. And remember, you got 2 copies of every chromosome except for the sex chromosomes. Once these are made, they're going to cut this other chromosome. And then homology-directed repair is going to mean that it's going to jump from one chromosome to the second one. So a thing that starts out just on a little circle of DNA, a little plasmid, is going to end up being on one chromosome and then eventually on both chromosomes. And then when this individual goes on to have offspring, it's going to transmit one of these chromosomes to its offspring. And it doesn't matter which one, because they both have the gene drive in there. So that's a gene drive. It's a kind of a self-replicating CRISPR-Cas9 system, which is, pretty neat technology, but also kind of scary technology, because this is, going to spread through a population, potentially very quickly. And so this has been used to genetically modify animals. And normal inheritance says that you'd have a particular allele and half your offspring would have it. Great. Then you'd have a particular allele and maybe half your offspring have it over here, and so we got a little bit unlucky. And then you get a particular allele, and we're going to carry this. And so at each stage, the frequency of the gene is not really going to spread in the population. But with a gene drive, you're going to mate, and at least half of the offspring are going to have this. And then as the offspring go to mate, they are going to eventually end up spreading the gene throughout the entire population. Because the gene will always be inherited, because, even if it is on the Y chromosome and just goes through the males, every single male that's born is going to have a Y chromosome with the modification on it, and it will spread through the population. And if you put it on a different chromosome, one of the autosomes, chromosome 6, for example, then you can actually spread this thing even faster through males and females. And that's a gene drive. That's the idea behind a gene drive. And there are lots of ways that gene drives can be used. In the next lecture, we're going to focus on a way that is aimed at mosquitoes. And these are mosquitoes that carry different diseases, including malaria and Zika virus. And so we'll see how we can potentially use and how people have used this to target those, at least in laboratory experiments and potentially also in the wild. I think this is something that is very much worth thinking about. And there are a lot of other people out there certainly thinking about this. It's a technology that I think will either become common or will be banned in your lifetime. And I don't know about banning it because you can do that in one country, but you can't do it everywhere. And mosquitoes don't know about national borders. So, yeah, essentially, this is something that anybody can do with relatively cheap technology. It's probably less than $300 to custom synthesize yourself a guide RNA and a Cas9 gene, and make a little plasmid that you can use to make a gene drive. So, some of the other interesting technologies have actually been used experimentally against mammals. And you can use these for population control. So one of these is a technique called a Y chromosome with an X shredder. So this is a Y chromosome that's going to have a gene drive, and it's going to make a CRISPR Cas9 system that will cut the X chromosome. And you can't live with just a Y chromosome, guys. Yeah, you got to have an X chromosome there too, because the Y chromosome really does not add much to you genetically. Yeah, hate to say it. And so with one of these, which is an X chromosome shredder, the X shredder approach, you can actually kill off all the males in the population. Yeah. Ugh. There are also ways to employ this that would kill off all the females in a population. Or there are ways to pass along genes that would make all the males sterile or make all the females sterile. There are, a lot of clever ways to use this that can have long-term consequences. And potentially, if you do it right or do it wrong, depending on your point of view, you can even drive a species all the way to extinction. And so here we're running into, I would say, the field of bioethics. So there are a lot of things that are possible in biology, but certainly not everything is ethical. And this is kind of outside this course. The point of this course is to show you the tools and how they work. Bioethics is its own field, and unfortunately, I am probably not qualified to actually walk you through a bioethics course, because ethics are going to differ depending on the time, depending on who you are, how you grew up, depending on the national mood, depending on a lot of things, depending on the economy to some extent. And so from time to time and place to place, ethics are going to vary. And I think it's a scientist's job to be ready. When the ethics are right and when the world needs help, you've got to be there and be able to say, here, this is how we can solve the problem. But when the time is wrong and the ethicists tell you, don't you dare do that, you got to find something else to do, because this is, eventually, all science is done in society. And so if you are going to go on in medicine or in science or basically in any field, you're going to end up with everything you do being connected to other people, whether you want it to be or not. Universities are publicly funded or privately funded by students. Students feel as though they want to pay for certain things and not other things. And you can understand, right, as students. Same thing in the world. If you are getting government grants for your research, depending on who's in power, who's in the White House or who's running the NIH or whatever, organization you're getting your funding from, even the Bill Gates Fund, they're going to have their own ethical criteria and certain kinds of experiments, no matter how scientifically interesting they might be, are just not going to be fundable because they're going to run into ethical problems or ethical concerns. So, interesting thing to think about, and something that I think, you've kind of got to come up with the answer to that on your own. You got to decide what works for you, And then ultimately, you've got to kind of take the temperature of the community around you, because you're not doing science in a vacuum, and you're not doing medicine or doing human genetic research in a vacuum. You live in a town that's you're part of a community, like it or not. And so, yeah, you've gotta. We normally have committees that would look at the bioethics before you're allowed to undertake any particular experiment. And they're made up of partly community members and partly medical professionals and partly scientists, other scientists who don't have a particular iron in the fire or a dog in the fight. That's a pretty good way to do it. But even then, if you got the right or wrong mix of people, they can come to decisions that may not be representative of what you would want or what somebody else would want. And so, yeah, that's a concern. But anyway, enough about ethics, because, like I said, I am not qualified to really do anything related to ethics here. And so I'd say those last couple of slides, don't worry about those. They're just looking at other neat things that we can do with DNA with a gene drive sort of system. So thanks very much. And the next lecture will be on gene drives and mosquitoes.